Hi, I'm Rishabh Chatterjee, and I'm presenting on behalf of the Research Informatics team at City of Hope. And I'm going to be talking about usual questions with unusual answers, application of multi-class supervised algorithms to identify COVID-19 viral strains. So here is a quick outline of what I'm going to be discussing today. I'll start with multi-class classification, move on to some methods of how to reduce that into a binary classification problem. We'll also talk about some objectives about how we use these techniques on a problem in biology. We'll talk about the feature engineering associated with that project, some preliminary results, and we'll also have some conclusions and future directions. So I'd like to start off by talking a little bit about multi-class classification. So the first thing to keep in mind is that it is a type of classification problem where instances are separated into one of three or more classes. And we can see in the visualization on the right side that we have three different clusters with different color codes. And each of these clusters here are supposed to represent a particular class. And so essentially, the goal of our machine learning algorithm is to try and separate these three points, types of points from one another as accurately as possible. And we would do this in this supervised setting. And another thing to keep in mind is that there are actually fewer natural multi-class classifiers than there are binary classifiers. And that multi-class classification is actually more difficult to optimize than binary classification is. And this can be due to a multitude of situations. In particular, class imbalance is actually a huge reason for this, because instead of looking at the imbalance between two classes, you're looking at the imbalance between multiple classes. And the other thing to keep in mind is that there could be a lot more limitations when it comes to the data that you have for training. It would be best if you had more data in order to expect decent results from a multi-class classification problem. So when we're given these kinds of situations, and especially in a setting like biology, you would expect that you don't have that much data to begin with. So how can we deal with some of these issues of multi-class classification? So one of the things that we can do is we can transform it into a binary problem. And the particular term in machine learning theory for this kind of task is called a task reduction, which just means that you're trying to transform the multi-class problem into several binary classifiers. And there are several strategies to do this. I'll first start by talking about the two most common strategies, and then I'll talk about two more approaches which are more new and also have some interesting benefits. So the first two strategies are called one versus all, which is also known as one versus rest, and the other strategy is called one versus one. So if we look on the left side of this slide, we can see in the one versus all that you have three different classes. We have green, blue, and red. And essentially what we do is we create three new binary classifiers. The first binary classifier takes the green class and pits it against the red and blue classes together. And we call that class as rest or all. And similarly, we do a similar design with the blue class in the second classifier, which we can see here. And we also do the same thing with the red classifier here. And in the one versus one scenario, what we do is we look at what are the possible combinations of pairing these classes against one another. So for example, here we have these red squares that are being pit against these black diamonds. And then in the second design here, we have these blue dots being pit against the black diamonds. And then the last possible combination for this particular scenario is red squares versus the blue dots. And so when we have these two very unique designs, the first question that might arise is, well, how do we make decisions from these multiple classifiers? So let's start with the one versus rest scenario. So what happens is that each of these classes that we pit against the rest or all, these classes are actually the positive class in our experiments for the binary classifiers. And so what we do is we take the probability of that positive class. And so the probability of that positive class could come out for a particular instance, in this case, 0.3 for green. And in this classifier here, it could be 0.7 for blue. And in this classifier here, it could be 0.2 for the red. And so what we essentially do is when we get these probabilities of the positive class for each of these classifiers in one versus rest, we essentially just want to see which of these has the maximum positive probability. And so that maximum turns out for this instance to be 0.7. And so for an unknown instance, you would use this kind of a scheme in order to make a decision about your final prediction. Now, on the contrary, for one versus one, we have a different scenario. Instead of looking at maximum probabilities, what we do instead is we try to see which class is predicted the most times. So if we look at this first instance, you can either predict the instance to be a red square or a black diamond. So in this case, the instance is predicted as a red square. In the second design here, we can see that the blue dot is predicted instead of a black diamond. And in the third design, we can see that the red square is predicted as opposed to the blue dot. So when you have this scenario, we can see that based on all the combinations, that the red square was chosen the most times. And so because of that, we decide to go with the majority vote that our final prediction should be a red square. Now, based on these two strategies, there could be some challenges. In particular, let's say with one versus one, if you had 10 different classes, the number of combinations that you have would be drastically different than what you see here. In fact, it's going to be numerous. And when that happens, you have a more challenging scenario where being able to get a tally like this of seeing which class is predicted the most becomes more and more difficult. And so when it comes to greater scale of these classes, one versus one is usually not preferred in that scenario. And it's also a much slower approach when it comes to the computational time, because you have to compute so many binary classifiers. And for one versus rest, one of the limitations is in the sense where what if the classifier itself suffers from other problems like class imbalance, like heavy class imbalance, or what if it just is not very good at predicting that particular class? If that happens, then it's quite difficult to make an immediate decision as to whether we should trust these probability values of that positive class for making our final decision. And yes, there is also one other limitation for one versus one, in which you're actually not using your entire data set for the training sample. One thing that happens is, since you're comparing red squares versus black diamonds, you're actually only looking at those instances, and the blue dots are completely omitted. So that also reduces your number of training samples, and et cetera. And that can also lead to some other implicit biases for your experiment. So besides these two approaches, let's also talk about some of the newer methods for reduction to a binary classification. So this first approach is called the error correcting output codes, or ECOC. So this is a very drastically different take than what we have seen so far. And the way that it works is based on the number of labels you have. Let's say that you have 4 labels like we see on the left here, where you're looking at different document categories like politics, sports, business, and arts. And essentially what we're doing is we're creating something called a particular code. This code is a binary string, and a binary string made up of n bits. And this number could be arbitrary, it could be intentional. But one thing that you have to keep in mind is that when you create this binary string, that is supposed to be uniquely represented for a particular label, it has to be drastically different from the other labels. That's the first requirement. The other requirement is that when you are positioning them against each other, these rows, they have to, these particular values here, they should be far apart. Some of them should be far apart. And usually what we like to say is when it comes to this situation, that each of these bits, these positional bits here, we say that we're going to be creating a binary classifier for each of these bits. So let's say you look at this first scenario here, where all the zeros are going to be corresponding to class 1, and all the ones are going to be grouped together as class 2. So that means that this first classifier has to pit politics and sports against business and arts. And so in this next one here, we would see that politics would be opposing sports, business, and arts. And so this is how you can get a distinct binary classifier for each of these different bits. And so that's one thing to keep in mind. That's how you get these distinct classifiers. The other thing to also keep in mind is that you have to make sure that you do have a contrast in the actual bits. So you have to have zeros and ones. If you have all zeros in one column, then that column can't be used to create a classifier because there's nothing to distinguish. Another thing that's important is, how do you get a decision from a prediction for a new instance in this scenario? So what you do is, since you're getting a classifier for each of these columns here, you would essentially take each classifier and get the features of your instance into each of these classifiers in order to create a new bit string. In this case, it's a 10-bit string. And essentially what you want to do is you want to compute the Hamming distance of this particular binary string of your prediction with these unique binary strings that correspond to the label. And what you want to do is you want to see which one gives you the minimum Hamming distance. You can see that I've computed these here. And this first one gives you a distance of 1, which is the lowest of these 4 digits. So therefore, our final prediction for this particular instance would be politics. So in the next approach that we're going to be talking about is called loss-based decoding. Loss-based decoding is a very interesting approach because we are essentially trying to figure out a way to unify the best of one versus of these binary classifiers, as we've seen before, as well as the this ECOC. Like, how can we unify them to get something more substantial? And so one of the ways that we can do that is by taking an approach where let's recreate this matrix, but instead of having just binary strings, we're actually going to put negative, 0, or positive. And these are going to be in a unique distinction from one another for each class. And now what you want to do is make sure that you still have contrast. You want to be able to say that negatives can be pit against positives, and zeros are just kept neutral, so that class wouldn't be counted as a part of the binary classifier. So if you look at class 1 here, or sorry, not class 1, but column 1 here, you can see that green is given 0, and that our yellow is negative, red is positive, and blue is positive. So that means that red and blue would be pit against yellow in this first binary classifier. And in the second one, we can see a different story where green and yellow are pit against blue. So this way you can get unique binary classifiers for each of these columns where you have the negative, 0, positive matrix. The other thing to keep in mind is you might be wondering, what are these values that you see here? So these values that you see here are actually talking about different margins. So one very interesting thing about this approach is it only works for margin-based classifiers. So essentially, we want to get a real valued output from a decision boundary function of our classifier. And so that real valued output is essentially what we are getting here for a particular instance and based on these classifiers. And so with these margins, now what we want to do is based on the classifier that gave us these margins, we want to be able to use the loss function of that specific margin. So let's say we're working with a support vector machine. We would define our loss function as the max of 1 minus z, where z happens to be our margin, our real valued output. And so we can also do this with C4.5, which is using a decision tree for classification. And you can also do this for logistic regression, AdaBoost, and these are some examples of the loss functions for least squares regression and CART. So one thing to keep in mind here is that depending on your classifier, the results that you get can be drastically different. So now let's say that you have this design, and now let's say that you have these margins. So what we do is we look at these classes row-wise, and what we do is these values that you see here, where if it's a 0, then 0 goes into the loss function as the input. And if it's a negative, we take the negative of the corresponding margin of that column. And if it's a positive, then you take the positive corresponding to the margin. So let's say we do this for the green. So you get loss 0, and then you look at this next column here. So it says over here that it's a negative, so you want to negate this particular margin. So that's why it's a positive 3.1 instead of a negative 3.1. And then we do 0, and then we negate the 0.7 because it's negative here. And then the positive is going to be the 6.2. So when you do this, let's say that this is for a support vector machine, you may end up with this value here of 3.7. And so we would do this design for all of the classes, for all of the columns, to compute the total loss for each of these classes. And when you do that, the one that gives you the lowest loss is the one that you would choose as your final as your final prediction for the instance. So in this case, 3.1 is the lowest loss. So that means the yellow class is going to be our final prediction. So now let's talk about a little bit of application here. So we are going to be trying to classify SARS-CoV-2 sequences in terms of taxonomy, their viral taxonomy. So within biology, organisms can be classified at different levels of taxonomy. And so there are some levels in which it's easier to classify them and some that are more difficult. So we actually have a pyramid that allows us to determine which ones are more complex and which ones are not so complex. So what we're seeing here is we're looking from the most complex to the least complex, where realm is the most complex and subgenus is the least complex. And what you're seeing in this highlighted blue here is what the SARS-CoV-2 virus should be classified as at each of these taxonomic levels. And you can see here that viruses in particular have different sub-levels within these different taxonomic levels. So we can see here it should be ribaviria, and then at the lower end, let's say with subgenus, it should be cerbecovirus, and then at the genus level, it should be beta coronavirus. And so essentially what we want to do is we want to create a classification problem where just given the genetic sequence of our viruses, can we predict the proper sublevel at each of these taxonomic levels? And so here's a quick machine learning workflow of how we've done this. And for example, we've just taken one specific taxonomic level, the genus, which has 4 different sublevels. It has delta coronavirus, alpha, gamma, and beta. And I'll discuss the features we used in the next slide. But we essentially use a 10-fold cross-validation approach. We generate several classification models to assess performance. And then our metric that we want to optimize on is the percentage accuracy of a holdout set of SARS-CoV-2 sequences, where we want to see out of a set of 200 SARS-CoV-2 sequences, how many of them are we classifying correctly. And also the other thing is in terms of our limited data, we actually have 50 different sequences for each of these different sublevels at each taxonomic level. Okay, so the features that we have used are the discrete Fourier transform and Shannon's entropy. So just to briefly talk about how we're using the discrete Fourier transform here, is we create a numerical representation of our genetic sequence. And then based on this numerical representation, we feed it into a discrete Fourier transform and compute the average magnitude of that output. And that average magnitude becomes our final feature based on this paradigm. And then for Shannon's entropy, we essentially are just looking at frequencies of our nucleotides, adenine, thymine, cytosine, and guanine, and we are computing their probabilities with their log 2 probabilities and just negating the final summation. And in doing so, we're finding the measure of the intrinsic uncertainty which is embedded within that genetic sequence. And so after applying these two types of features, one thing I forgot to mention here is how did we go from the genetic sequence to the numerical sequence? The way we have done that is based on several conversion rules. And these conversion rules are inspired by another study by Randhawa et al. And essentially, these conversion rules are saying, how do you map each nucleotide to being numeric? So in this case, we have something called purine and pyrimidine, where purines are given a number and pyrimidines are given the other number. In this case, it's very binary. So adenine and guanine are given 0, and thymine and cytosine are given 1. And then there are also other different representations that you can do. But we have chosen this particular conversion rule because we've seen that across several taxonomic levels, we're getting much better performance with this particular conversion rule. And here is just a quick outline of the feature engineering process, because we do have a third feature which we've actually chosen as our final feature for the classification model. And the way we have done that is we compute the averaged magnitude of the discrete Fourier transform. We also compute the Shannon's entropy. And then after doing this for a particular sequence, you divide the average magnitude by the Shannon's entropy. And this ratio of them is called magentropy. And this magentropy becomes the final feature for each genetic sequence that we put into our machine learning experiment. And here are some preliminary results in which we particularly are looking at our holdout set of SARS-CoV-2. As you can see in this bar chart here, that we have applied this experiment to all the different taxonomic levels. And we can see that for the most complex level, the realm level, we're having a huge challenge with trying to predict SARS-CoV-2 as ribaviria. In fact, only 33% of those 200 sequences are being predicted properly as ribaviria. And then if we look at these other different categories here, these different taxonomic levels, we can see that it predicts all of these 200 SARS-CoV-2 sequences as the proper labels at those different taxonomic levels, as the proper sublevel. And at the genus, we can see that 97% of those sequences are predicted as the beta coronavirus. And just a quick thing here in terms of the consistently best performing classifiers, this was done using extreme gradient boosting trees and decision trees. We actually used PyCaret, so we have assessed several models in a machine learning catalog for doing a comparison at all these levels. And we found that these two were the consistently best performers. Also, another thing to keep in mind is the experimental design that was done here is that it was actually based on the multiclass classification on its own. That means that the decision boundaries can naturally handle multiclass classification for some of the models, and then it chose one versus rest for the models that do not have that native capability for PyCaret. And so one thing that we have done is we wanted to assess what if we did one versus rest and one versus one for just one particular taxonomic level. We wanted to do a compare and contrast with what we see here. And so here's some results of that, where out of all the models in PyCaret, we have ensured that we have employed the one versus rest and one versus one strategies for the entire model catalog. And what we've seen is that for the light gradient boosting machine, the one versus rest gives us the best performance in terms of our average 10-fold cross-validated accuracy. We also get a standard deviation which is slightly above 5% across all the folds. And on the testing set, which is called the holdout in this case, the testing set for that particular experiment in PyCaret, where we did 70% for training and 30% for testing, that 30% testing accuracy is actually 85.25%. And the other thing to keep in mind is with the holdout set, which only contains the SARS-CoV-2 sequences, which are the 200 sequences, we are ending up getting all of those sequences properly predicted. And then in our one versus one design, we can see that our 10-fold cross-validated accuracy is much lower than one versus rest. And similarly, we have a higher standard deviation in one versus one across the folds. And then our holdout accuracy is lower, but yet, even with these lower numbers, we are seeing that the SARS-CoV-2 sequences are still being completely accurately predicted. And so when you see results like this, it makes you question, which method, when you do the reduction from multiclass to binary, is the best method to use. In this case, it really depends on the objective, I would say. But if we were to look at this in terms of trying to classify all the sublevels, based on just these two experiments, I would say that one versus rest is the better choice. And we do plan to do some further experimentation on other levels to see how this comparison between one versus rest and one versus one actually holds out. And just some preliminary conclusions and future directions to keep in mind is that, as I mentioned, the choice of the multi-class to binary design is very problem dependent. When we talked about one versus one and one versus rest, we could see that in this case, we had a problem with only 4 classes. And so in that situation, we saw better performance at one taxonomic level, which was genus, for one versus rest. However, that doesn't mean that it's necessarily going to hold true at other levels. And the other thing to keep in mind is that the magentropy feature, which is what we generate as our final feature, this feature is actually very predictive. It has a very high feature impact as opposed to its counterparts that make up the feature. And the other thing to keep in mind is that we have not used ECOC and loss-based decoding yet. And the reason for that is for these matrix-based approaches to work, we have to ensure that we have enough data. As I mentioned before, we're running very low on data. In fact, we only have 50 different sequences for each sublevel at each taxonomic level. And so that's something that we're trying to improve, where we get more examples of viral sequences at each of these sublevels. So then we can apply these techniques like ECOC and loss-based decoding. And one thing that I haven't mentioned is when would be the best time to use those particular algorithms? So what I've seen in general is that loss-based decoding usually outperforms ECOC. But one thing to keep in mind about it is it only works well when you have a large number of classes. When you have, like, let's say 8 classes or higher. That's when loss-based decoding will probably outperform these other approaches. But when you have less classes and limited data, it's possible that in some situations, loss-based decoding may still be the victor, But in other cases, it may go down to ECOC or one versus rest. So there's really no perfect way to explain which will be the winner in terms of the design. However, it's important to compare and contrast several designs based on your problem, as it's very context dependent. And the other thing about ECOC and loss-based decoding, which is a very huge weakness, I would say, behind these two algorithms, is the way that you construct your matrix is crucial. In fact, some of these matrices can be created through different rules, like hill climbing algorithms and etc. But in other situations, it's actually very context dependent. In fact, in the original paper that talks about ECOC, there's actually different criteria about the different digits in the MNIST data set, which are used to decide how many different bits you'll have for your matrix. And so what we've seen is that when it's more context dependent, ECOC tends to perform better, and maybe even better than loss-based decoding. So there's a lot of different things to think about when it comes to using these different reduction techniques. The last two things I want to mention are that the methods that are developed in this experiment are general enough to be applied to genomic sequences from any organism. We're not limited to humans. We can do this for bacteria or any organism. And finally, this low computational cost approach can predict any viral taxonomy, and it can definitely be used to prevent the next big pandemic. We still have a lot of testing and experiments to go, but we're very, very confident that we have a solid approach for doing so. And finally, I just wanted to acknowledge my colleagues at City of Hope. Firstly, I'd like to thank Anushka Bhatt and Isha Anand, who are two interns that I mentored over the summer and spent a tremendous amount of time with me in creating computations for these experiments. And I would also like to give my heartfelt gratitude to my mentor and supervisor, Dr. Sri Sairam Achuthan, and also the directors of research informatics, Samir Kordi, and the chief informatics officer, Serena Nadav. So thank you very much for your time, and we are now ready for Q&A.